Good morning, everyone. I am Teresa Przytycka from National Institute of Health. The institute that I am affiliated with is National Library of Medicine, and I forgot probably to press something here. How about now? Better. Thank you. I am Teresa Przytycka from National Library of Medicine, which is part of National Institute of Health. And I am a PI leading a small group whose research is focused on developing algorithmic methods in general in computational biology. Through the years, we've been focusing on various areas for which we have been looking at developing methods and algorithms. And for the last couple of years, quite a bit of our research have been focusing on mutational signatures. And today I am going to present basically a tutorial to kind of introduce to you the subject, the computational challenges that emerge in it, and a little bit also information what we as the group have done in this area. As you are perhaps well aware that DNA in every single cell is subject of constant damage. So that damage may be, even if the optimal environment, it may be related to the environmental factors such as UV light, the reactive oxygen species that we are surrounded with, and may be also imposed by smoking and drugs. It has been actually estimated that on average, each DNA in every cell may receive as many as 10 to the 5 lesions per cell per day. As also, just to add to that, as another important component to the DNA damage are just natural processes that are within the organisms, such as replication, which are really also not, which are also error-prone and are also source of DNA damage. So that would be really dramatic if we had, so many mutations per cell per day. Fortunately, cells also develop a very robust DNA repair mechanisms that are very robust, that are very redundant, and really most of those DNA lesions are being very efficiently repaired, and only a small fractions may remain unrepaired and then turn into mutations. So the cells depend on this balance between how much damage may happen and how efficient DNA repair mechanism is. If an organism is subjected to some really higher amount of the DNA damage, be that in the environment that is particularly unfriendly, too much of UV light, maybe too much of exposure to smoking, etc. So then the mutation burden, the lesion burden will be really high, and DNA repair may not keep up with it. On the other hand, also, it's possible that a DNA repair mechanism in a given individual is in some way weakened. It may be because germinal mutations or some other random factors, and then that will also cause a leakage of those unrepaired lesions and the increased mutational burden. So why we care about it? Well, it has been actually observed that often specific mutagen leave specific mutational patterns in DNA. For example, UV light is known to mutate C to T, but those mutations, C to T, are context dependent and are really rely on the fact that the C is preceded to T. So if the nucleotide before C is T, then that provides much higher probability for the UV light-induced mutation than C when it's in the other context. So there have been, UV light is not the only process that has this kind of characteristic mutational pattern. An idea is, can we take those patterns, read those patterns from cancer genomes, and use those patterns to understand the processes that potentially have led to a given mutational mutations in the genome. So to do that, the first thing that we need to do is somehow define what does it mean to be a pattern. So there may be many ways to define such patterns, but much of the really recent research in the area is utilize the definition of a pattern, the so-called mutational signature, that has been introduced by Alexandrov et al. In 2013. So their definition of the pattern is very simple. Specifically, they say, okay, let's divide all the mutation in the genome into categories. And then we are going to count the number of mutations in each category that will be associated with a given mutagen. So how we will define the category? So again, following the definition of Alexandrov et al. Say, well, since we know that the mutation might be context dependent, our category will be a triple of nucleotides, such that the central nucleotide is the one that's mutated, and then the flanking nucleotide provides the context. Since in double-stranded DNA, we don't know whether, say, mutation is from C to A or G to T, so there are only 6 different mutation categories for the middle residual, and then we have all possible flanking nucleotides, which gives us with 96 category. And the idea the mutational signature is simply defined as a distribution of the mutation over this 96 category. So what you see here is signature, I will come back to those numbers, a signature for the UV light, when you can see this concentration of C to T mutation, and this, you may not see it from the distance, but this little island here corresponds to all the categories where you have TC and then something else. So there are some examples of other, signatures defined using the same idea. Very important in cancer genomic ApoBec signature. So ApoBec is an enzyme. It's actually a family of enzymes, but some of those enzymes are known to mutate a single-stranded DNA. And then you see, again, the mutation, the red mutation from C to T, but you see that the pattern of the mutation is quite different and the distribution is quite different. Again, some other examples you can see below you have mutations that are associated with cigarette smoking, C2A mutation, and some example of chemotherapy drug. So we can have, so we would like to be able to really look into the genome from the perspective of these mutational signatures. And one important thing is that really we don't know all of the mutagen we have in the universe. We just don't know. We have some examples, but we don't know what else may be mutagenic, what other chemicals our body may be exposed, and so on. So we don't know that. But once we have a definition of mutational signature, a specific formal definition of a pattern, we can at least ask the question, given that this is how we define the mutational signature, and given the catalog of all the cancer genomes, what are the mutational signatures that are needed to explain all those mutations in the individuals that we see in the databases? So why we do care about this? First of all, of course, we would like to understand the mutagenic processes in human organisms. So that's basic knowledge. We like to know what processes causes mutation, and eventually lead to cancer. So that's first. But secondly, we'd like to understand them better from the perspective of preventions and diagnosis. So for example, if we observe that certain group of individual, maybe geographically somehow related, display some new mutational signatures, then that give us a clue that there is something in the environment of those individuals that creates a mutation. We would like to identify this mutagen and then be able to say, okay, stay away from whatever this is substance that you might be eating. So that's certainly one example of that. And the last very important application, so the first application is related, what caused the mutation. But the second reason for which we can have this leakage of the lesion, unrepaired lesion, is related to the fact that DNA repair mechanism may be not efficient and even broken. One famous example is in breast cancer. Many patients experience homologous recombination deficiency, so the DNA repair mechanism that's known as homologous recombination repair is broken. If we know that cancer cells have certain repair mechanism broken and there's enough understanding of the DNA repair mechanism, the idea would be to look at what is the complementary DNA repair pathways that allow the cancer cell to bypass this problem. And then we hit this complementary pathway with a drug, and we selectively kill cancer cells via the process that we refer to synthetic lethality. So one example, again going to homologous recombination deficiency, are PRP inhibitors, which inhibit complementary pathways, which allow the cancer cells that have the deficiency in one pathway to disable other pathway, and then the cells cannot replicate. Okay, so this is motivation. So here is like as for the general overview of computational problems that we have. Sorry, I don't have a pointer, which makes it a little bit difficult, but let me deal with it. So basically, we have those mutations that are caused either by problems with the DNA repair or by environment. And then we came up, what we have to do is somehow define the mutation categories. I show you one example, but that's an open question. What are the most beneficial categories? And then being able to computationally identify those mutational signatures. Once we infer those mutational signatures, there's a couple of course, statistical questions about stability of those signatures, etc. But once we have those signatures, this pattern, we would like to know what is their relation with either external world or with linking those signatures to molecular causes, to their role in cancer, maybe also in gene evolution and diversity. So I'll start with the mentioning the first problem: how do we come and how we, having defined what a mutational signature is, how we can identify mutational signatures that are required to explain the mutation in the cancer genomes that we have in databases. So again, this is following the paper, the same paper from Alexandrov et al. From 2013. So basically, they propose, we'll use non-negative matrix factorization. So in the red matrix or orange matrix is the matrix of all the cancer patients or cancer data. Every row is a sample. Every column is mutation category. So we have 96 of them. We have for every entry is the count of the mutation of a given category in a given sample. And our goal is now to identify those signatures that I defined previously as those vectors of length 96 and for the probability distribution of a given mutation category times the what we call exposures. So that the mutations in each patient are being explained as linear combination of those signatures times those exposure, those linear coefficients. And the exposure is actually a good name because it's been shown for at least some examples where we understand what the relation with the environment is. So for example, it's clear that for the signatures that is known to be related with smoking, the exposure indeed is related with how much given person smoked. So it is in some way we interpreted that this is an exposure to a given mutagen. Okay, so using this technique, we have there's a database of mutational signature delivered in this way. This is cosmic mutational signature. So when I show some numbers, those are numbers of those signatures in the database. You can look through them. If you zoom to this web page, you can kind of quickly see a summary what we know about the signature and what is not known. And this class is probably more half empty than half full. We know a little bit about many of those signatures, but we don't know a lot about most of them. So one question would be how we are going to link those signatures to some molecular causes. However, before I move further on, since we are going to talk about computational challenges, so assume that we have now all this mutational signature being defined. So if you have a new patient, you would like to ask the question, okay, what are the mutational signatures that are present in the genome of a given individual? So this we call it this refitting problem. This is a problem when we do not try to find a new mutation, but simply try to find which mutational signature are present and with what strength. As trivial as this may be, it's actually not so simple. And mostly it's not so simple because as research continues, the number of these mutational signatures increases, and currently we really, if we just, it's about more than 100 of them, and problem starts to be not so well defined. So usually in this question, when we ask, what is the strength of mutational signatures for a given individual, we usually would like to know what sort of cancer that person has. And for most of the cancers, we have research based on a larger cohort that say, okay, breast cancer, typical for breast cancer are those signatures. Okay, so that's one. There's again, given that there's many programs that have been developed to solve this problem, to fit these mutational signatures. One have to be aware on how stable those signatures are. So this is just using quadratic programming that will allow us to find optimal decomposition to those signatures. We use bootstrapping to see how stable those signatures are. And then you can see that some signatures are very, very stable. So after bootstrapping distribution is not very broad. However, some other signature, like signature 5, are more noisy, and those are more challenging to discuss. And when you look at the distribution, those more noisy signatures typically correspond to the distributions that are more flat. So that's one challenge. Okay, now let me move to the second group of challenges. Assume that we have those mutational signatures, but as I say, for only very few of them, we understand quite well what they are doing, and for most of them, our understanding is incomplete. So I'd like to be able to have a better understanding of those signatures. So perhaps the first thing that we would like to do is to be able to look at the mutations in the individuals, even assuming that we know what mutation processes are in those individuals, to assign this mutation to the mutagenic process. So one way to do it will be simply, look at, okay, if we have a T to C mutation in this context, which signatures is most likely to produce it, and also which signature is most frequent in a given genome, and have a simple model like that. However, we can appreciate that if you look at the sequence, the mutation signatures that hit consecutive position or close nearby position might not be independent. So this is one way, and this is indeed the case that, for example, ApoBec signatures requires that the DNA is single-stranded, and if DNA is single-stranded, it's really usual on some extended region. So in this approach, we simply develop a hidden Markov model that we will model dependency here between mutations that are nearby in the sequence, but using independent model for if the mutations are a certain distance apart. So we call those first group as a clustered mutation, or sometimes clouds, and the second group mutation as dispersed or sky mutation. So those two mutations groups, the dense one and the sparse, have different properties. So for example, here is showing one of the properties is that when we look at the probability of occurrence of a given mutation from a given signatures as the function of replication time. And for people for the general audience, I would just wanted to say that if cell replicates, the replicator starts simultaneously in several regions and then proceeds. And then what we see here is that really the probability of the mutation depends whether a given region is being replicated earlier or later. So you can see here that dense and sparse mutations have often different property with respect to replication time. So that's some of the insights that we obtain this way. There are other questions that been asked, related to say which mutational signature are more likely to hit nucleosome-free regions, which are more likely to hit nucleosomes, which are more likely to hit regions that are actively transcribed, and the regions that are not transcribed. So those are all these differences that once we have those clear definition of mutational signature have been very, very actively explored. One of the things, for example, that we notice here is the signature that 2 and 13 that are two ApoBec signatures, so those signatures that are known to be caused by this specific enzyme. There are two different signatures, and the reason is that the damage is the same, but it's repaired into different ways. So it's leading that you have almost the same pattern, but repaired in a different way. And we can indeed confirm here that those signatures, the exposure of those signatures is related, at least for the signature 13, is very strongly related to ApoBec expression, as you would expect, and that correlation is not so strong for signature 2, especially considering the variant for the dispersed signature. So in this way, we can capture the dependency of the exposure of the signature on the gene expression. And this is interesting, and we would like to be able to capture more of those, since this is just a quick review, or maybe I'm not going to go into it, but to remind that there are many, as I said, the cells develop many different DNA repair pathways, and here I just list some of those pathways that are specialized to different type of damages. So indeed, if we could see that strength for some signature is related to increased expression on increased activity of one of those pathways, that would be very useful for us and will help us to link this signature to those repair pathways. So that's motivations, that we really would like to know the relation of the signature activity with the gene expression activity. So of course, the simple thing what we could do is just run simple correlation. So here is one idea. We can, for many cancer genomes, we have information not only about the mutation in the genome, but also in gene expression. So for those, when we have both of this data, we can construct those two matrices. One is a matrix of gene expression, the green matrix, patient by genes. The second is a matrix of signature exposure, will be a patient by signatures. And for every pair, gene and signature, we can look if there is a correlation here. If there is a correlation, we can kind of save this information of this gene by signature matrix, where we keep only the genes that correlate with at least one signature. And at the end, what we could do is we cluster the genes that have the same correlation patterns with the signatures. Once we do that, we can simply do enrichment analysis and try to see if there are any patterns, any insights that we can get from this clustering. So this type of, as example of the study for breast cancer, again, a glass half empty or half full, how you would like to see it. So at the bottom you have each column correspond to the signature. At the top I gave the name of the signatures where we know something about it. In particular, we have two signatures that we refer to as the clock signatures because they've been identified as the mutation in those associated with those signature is correlated with patient age. And one nice insight that you can see from this analysis is if you have the first clock signature and the second clock signature, you can see that the second clock signature correlates with the cell cycle. So in fact, we actually, in this case, the signature is quite well understood. So it's consistent that those, indeed, these signatures accumulated at every replication. So the signature 1 over there counts the replication time. However, we know that signature 5 doesn't do that. This is a signature that's not fully understood. The conjecture is that that signature is somehow related to some very common environmental exposure and also accumulated with age, but not necessarily with cell cycle. And then in the center, this is the not so informative. We have a couple of signatures. Some of them we understand, and the one with the question mark we don't. And from this clustering, we really cannot tell it from the remaining signatures. So this is not informative enough to tell us anything about signature 8. So the next, perhaps, idea would be, everything's, you have so many correlation of the gene expression in cancer genome as we can appreciate. So let's do something more informative, or maybe that can provide more detailed information. So let's use partial correlations. So here we would like to say, okay, if let us consider two type of entities, genes and signatures. Genes are here in green, signatures are in orange. We know that whether it's a gene and signature, we can associate with it an activity. Activity of gene is gene expression, activity of signature is strength. So we can play with the correlation, and this is in fact what we've done in the previous slide, but now rather than looking at the correlation, we look at the partial correlation, and in additional add the request assumption that those correlations need to be sparse. So basically, practically what we do is for every either gene of the signature, we ask, what is the sparse, the small number of other items that can explain a given activity of a given node as a linear combination of the activities of other nodes. So this will give us the sparse networks. It's directed because it's asymmetric. We have additional statistical methods to direct some of the edges that are unresolved and have direction in both into bidirectional. The direction at the end didn't appear to be all that important, but sparsification was very helpful. So looking at the same data now with this more focus, more approach, we can appreciate it now that when you look at what are the genes that are associated with this unknown signature 8, we know that this is nucleotide excision repair. We have also over there, when we have these apobec signatures, we can appreciate that they are associated with immune response, and in particular with the positive regulation of regulatory T-cell differentiation, which are related also to immune checkpoints. So we start to gaining a little bit more detailed information. Nevertheless, that's confirmation of this is subject to experiment, and some of the, indeed there's experiment confirming that the signature is related to the specific pathways. So we have some. So that's one way we to learn about what those signatures might be doing, trying to associate them with the expression. The second way would be to try to associate those signatures with the mutations in, say, cancer driver genes, or some maybe other cancer genes. And the motivation for that, so for example, signature 3, the homologous recombination signature, is in part also can be caused by mutation in BRCA1. It's been observed that signature 5 appears in the cancers that have mutation in this nucleotide excision repair protein, ERCC2. So that suggests us that we perhaps should consider associating those mutational signatures with perhaps mutated genes or more broadly, say, mutated subnetworks. And this is another thing that we can do. So there are various methods. We ourselves developed method for the drug response when you identify mutated subnetwork associated with continuous phenotype. So without going into the details, you have this signature. For every patient, you have signature strength. For every patient, you have information what are the mutations. We have we superimpose those mutations on the protein-protein interaction network, and our goal is really to identify mutated subnetwork associated with a given phenotype. I'm not going to go too deeply into method, but just to tell what sort of machinery we use here. So we use integer linear programming optimization and building on a graph theoretical concept of connected set cover. Okay, what we get with that, again, we have with various mutational signature, we can now also associate mutations, and as we will hope to get, BRCA1, BRCA2 is associated with homologous recombination deficiency, which is signature 3, but in addition, TP53 is also related with these signatures. Up to also apobec, we can see again this associated with CHEK2, but also with the NOTCH1 pathways. I didn't point it in the previous slide when I was looking at the expression. Apobec was also associated with NOTCH pathways there as well, and other mutated genes. For some of them, they all kind of make sense, but of course, to really understand it more deeply, one will have to follow with experiments. Okay, now the next things which I would like to mention is those mutation signatures. Although I use them almost in the same way as gene expression for the network, they really didn't come like immediately. They accumulate over time. So the question one could ask is whether the mutagenic processes that act on a genome over time are always the same, or perhaps they may be changing with time. So, so this is a valid question. This is related to evolution of the mutational signature. How those mutation signatures evolve as the cancer evolve. So just to briefly, without going into details, briefly point that those indeed, those mutational signatures, do change. The mutagenic processes do change over time. So this is a study from consortium study on the evolution history of cancers, which in particular consider among how various type of processes in cancer may be changing in time. So the idea here, how you can put a time, how you can, given a genome, how you can reason about timing events. So there are characteristic events that you can put as things that happen before and after. One is whole genome duplication. So whole genome duplication, even if it happens, then looking at the mutation pattern, you can say which mutation happened before whole genome duplication and after. And the second thing is if you have also some evolution into subclonals, then you can also see which mutations are present in one clone or not in the other. So we don't have time per se, but we have some discrete events that we can say before and after. And then using this type of partition, what you see over here in the colors are this kind of general time regions. One can appreciate that SBS, this is for signature, that for colorectal cancer, they observe that signature 1, 5, and 18 is observed from the beginning, while some other signatures are being observed as entering to the picture later. And just looking at the subclonal evolution, this is from a paper of Mahmoud, right? You're here? Yeah, here. Ask him about it more. So this is from his group. They look at the subclonal evolution, and then you could see how knowing what the mutational profile of each subclone, they infer mutational signatures, and you can see that depending on which subclones you look at it, the mutational distribution of mutational signature is different. And this orange signature is the signature related to mismatch repair deficiency, and it's because in this subclone there's happened a mutation that hit this particular DNA repair pathways. So there's a lot of to think about it, about the relation of the time dependency of the signatures as the cancer evolve. Another interesting question is, some of the processes, such as smoking, are really, of course, they cause cancer, but they will do quite a bit of damage in the healthy individual. Can we somehow use this formalism or this concept of mutational signatures to look into what some of the mutagenic processes may do to healthy individual? And here, perhaps healthy is exaggerating, but I'll say in the non-cancer tissue. Getting signatures from anything else by cancer is not so easy, because the reason why we can get so nicely signature from cancer is because we have this expansion of one cell. So we have like the signal of where the mutations are to be replicated. While if you do a healthy tissue, you probably have to do single cell analysis. But there's a kind of middle ground that we decided to explore, which is like, what if we take control tissues? So let's take control tissue from cancer patients, and let's see what will take the cancer patients to read the strength of mutation signatures, and we'll look at the control tissue to see what happens in the control tissue in correlation with those signatures. So again, what we have here, we look at the gene expression, but then we decide to use Sarber sort, and understanding that in case of lung, a lot of things may be related to the reprogramming of the tissue's proportion in lung. So again, so here now we have those mutational signatures. I put what we know about it. Signature 1 is this first one that relates to cell cycle, so relates to tumor growth, probably not to environment. Signature 2 and 13 is apobec, immune. 5, we are not so sure. 4 is smoking, and 40 is very strongly correlated with smoking as well, so we will call both of them smoking. And now you can see how a relation here of all those signatures with the cell type composition, and those you have the names of the epithelial cells in cancers, and you can appreciate from this that smoking increased the number of goblet cells, but at the same time, it was also known that smoking also reduces the number of epithelial cells, which is this third from the bottom. So what we can see now from analysis, we fully agree with this observation, but we have some additional information that while smoking seems to have direct effect on increasing goblet cells, however, in terms of reducing of the say ciliated cells, we can see that's not so much smoking, but there is this other signature associated with it that is present in other environmental, not only smoking, as well as the immune signature that are associated with a reduction of those cell types. So we can use also an argument here that we can also use this concept of signatures to look into changes related to the environment, especially if you could have also the single cell sequencing data related when we can read the signatures from in single cells. Finally, we started with a definition of signatures using non-negative matrix factorization. What it basically tells us that we assume that all these processes are independent. That puts, and at the same time, the first analysis of mutational signature brought 20 signatures, next 50, and now we have over 100. So what's going on? Well, the mutational processes are not independent. And if we try to treat them as independent, we'll have this explosion of signatures that we observe. So at the beginning, I say that for mutation to happen, we may have mutagenic process and the DNA repair. Now, different combination of mutagenic processes and DNA repair deficiency will create different signatures. So we may have as many signatures as many of combination of those factors. So we have this explosion of signatures. So one way to deal with it is, somehow make a more complex model. So one that we propose, say, let's assume that we have two types of processes. We call them primary and secondary. And primary processes like UV light and whatnot will be independent, and we will dissect it using non-negative matrix factorization or similar procedure, while we assume that we have a second group of processes that really act on the primary processes, and basically those, rather than being causing a mutation, those processes fail to repair mutation with certain probabilities. So this makes the of course optimization problem more difficult, and presumably you would like to mix some of the knowledge so that you don't have to infer everything. However, surprisingly, it's even when you try to infer everything, it works quite well. So with that, I would like to summarize. I introduced to you this concept of mutational signatures that is being dependent on various choices. So one of it is choice of what's mutational category, another is choice what will be the way you infer those signatures, and also what sort of model you assume there's additive, non-additive model. So those are all, I think, computational challenges that are still very widely open to computational exploration, algorithmic exploration. There is also important, thanks to those mutational signatures, we already learned a lot about mutagenic processes in cancer and in genome. We learn about this non-additive, non-uniform distribution of the mutation types of the genome, for which we have increasing understanding why this is so non-linear. As I show, we can also look at joint analysis of this mutational signature and gene expression and mutated subnetwork to try to give some insight on what the signatures that we just observe as pattern may be, what may be biological relevance of those signatures. I showed that we can also use those mutational signatures to study environmental impact. Directly, we can study it based on the cancer genome, but with enough single-cell sequencing, we may be able to pick up mutational signatures that do not lead to cancer, immediately to cancer, and probe the environment, on the without assuming that the individual have to be in cancer state. We also, I didn't discuss it too much, but I also wanted to just re-emphasize that those signatures are also predictors of treatment option. These ApoBec signatures have been suggested as the predictive signatures for the immunotherapy that SBS3 is known signature for homologous recombination deficiency, and this is known that PRP inhibitor works well on those pathways, and we hope to see more and more as we understand what the signature means, what pathways they hit, and what may be the synthetic lethal pathway to hit with drugs. So with this, I would like to conclude and see if there are any questions.